Hello, everyone. We see a few people waiting or present in the stream already. Today's workshop is going to be on strawberry DNA extraction, and we're just going to wait a few more minutes so that more people can join.
Would it be a good time to introduce ourselves a little bit? I'm Ganna. I am a scientist, and I'm also a teacher. So I will be doing the experiments and talking a bit about DNA, what it looks like, how to make it. Yeah, and we also have Natasha in this room.
Hi, everyone. I'm Natasha. I'll be the moderator today. So any comments that you guys have, I'll be giving them up to Ghana, and I'll be controlling everything behind the scenes. Ghana, do you want to put up your slides now?
Yeah, sure.
And then once the slides are up before we start, I'll just do a quick territorial acknowledgement.
So can you see the slides now?
No. That's strange. It was just up.
It says it's processing.
Okay, we just need to have a few moments while it shows up. And people are still filing in, so no worries. We've got a couple minutes for this to pop up.
Well, it's still processing.
Hmm.
Let me see.
It's still not working. Pooja did just send me the slides, and I can try to share from my end.
Yeah, sure.
Our tech wizard is joining. Okay, here we are.
Okay, yay.
Perfect. And that brought us to 5 minutes. So we've left enough time for people to join. Thank you all for waiting. Hopefully we have some more people joining as we go, but I'm just going to start with a territorial acknowledgement. Oh, sorry, one moment. Everything is freezing. All right. So to get started, we would like to take a moment to reflect on our connection to the land and thank the traditional guardians of that land, which we at Squish Vancouver live and work. Vancouver is on the unceded, ancestral, and traditional lands of the Musqueam, Tsleil-Waututh, Stolo, Sechelt, Squamish, and more Indigenous peoples of Turtle Island. Some housekeeping. So my name is Natasha. I'll be the moderator today, and I'll be taking any questions through the chats. So if you have any questions, I will get them from there. So sorry, I've lost my notes. Okay, here we go. Hi, everyone. Welcome to today's workshop. This is the Strawberry DNA Extraction Workshop. And to get started, let me just go over some housekeeping. This session will be recorded and posted on YouTube after the live session. You and any of your students will not be recorded, so it will just be the content and the presenter. In today's workshop, we will be exploring DNA through a candy model of extraction and with a strawberry DNA extraction experiment. Our presenter, Ghana, will be taking us through all of this exciting content. So I will pass it over to you.
Yeah, thank you. Nice to meet everybody. So today we'll be doing DNA extraction, and we'll be making some DNA model with candies. First of all, let's talk a little bit about DNA. So one of the definitions is that DNA is the blueprint of life. And we have several sections for today's activities. First, once again, we'll talk about what DNA is. Then we will talk about how to make a DNA model using candies. And then we will actually extract DNA. And at the end, we are going to have some question time. So please pay attention because, we will try to go over everything we learned today. So now let's take, a 5-minute, let's call it break, and think about what is DNA, and try to write down everything we know about it before we go to the next steps, right? Okay, let's do it. We have 5 minutes, and take a deep breath. Get ready for the activity. Yeah, think about what is DNA, where DNA can be found, what DNA does, what is the function of DNA, anything related to the topic would help, right? Do you know what genes are? Do you know what chromosomes are? What part of the cell they make? You can see on the picture scientist who is kind of holding a DNA model, and think about what you know about it, right? What does DNA look like? So there is still a few more minutes. Yeah, and please feel free if you have any questions, please put them in the chat. I'll be more than happy to answer them. Okay, 2 more minutes. I'm pretty sure you've got a lot about DNA written down so far, or just thinking about it also helps. Okay, 1 more minute, and we'll be ready to move to the next step. Okay. Okay. So should be good to go to the next step. And what is DNA, right? It's a very important molecule in our body, and it is it can be found in all the living things. And if you look at the picture of the cell under the microscope, DNA can be found in the nucleus. And usually it is kind of packed into a chromosome, this kind of butterfly shaped body, right? And the DNA itself is a long strand of a double helix, which kind of coils into a chromosome. Okay. So DNA is actually quite a long molecule. Every cell of our body has, around 2 meters of DNA, but it's also very compact because it coils into a chromosome and it's very like assembled, right? And that's why it fits in the tiny nucleus of the cell, which you can only see under the microscope. And yeah, once again, DNA is a double helix, and you can only see it under the microscope. And today we will be making a model of DNA, and we will also extract enough DNA so that we can see it with the naked eye. So DNA is a double helix, which means that it has kind of, long rails, and it also has those steps. Sometimes DNA is also called a ladder, a twisted ladder, and you will actually see why it's called that once we make a model of it. So, candy DNA, our first activity. For this activity, we would need 2 of those Twizzlers. Okay. I'm gonna get 2. And we are going to also use some candies, right? And we also need some toothpicks to connect them all, to make the letters. Let's start with the rail, right? So we are going to attach some letters to it. And we are going to make just 5 letters. That's enough for our model. Right. So right now it looks a little bit like half a letter. Okay, and also I got some candies, and they actually represent nucleotides, and these nucleotides are shown in different colors, and there are 4 types of them. There is adenine, which is A for short, right? And we are going to make adenine in red, right? And then there is timidine, which is yellow, and it stands for T. And there is cytosine, which is green in our case. And there is guanine, which is kind of, dark purple, right? And there are certain rules how nucleotides pair up. For example, A always pair up with T. And there is an easy memo for this. Apple on the tree, right? So it's apple on the tree. And there is also another pair, like G and C. And the easy way to remember it is car in the garage, right? And we are going to use these rules while making our DNA model, right? So I'm going to start making our DNA sequence and using this half a ladder. So this is A, And then we have T. Okay. And then we have C. And G. Right? You see what's happening? And we are going to add another A because we want 5 nucleotides. Right? So we kind of made half of it. And now we are going to make another half. And we are going to use the same rules. So for adenine, we are going to use thymidine, right? AT, remember apple on the tree. And then against T, we are going to place another A. And this kind of pairing is called complementary. So right now we are making a complementary strand of DNA, right? So we are going to put G against C, right? And we are going to put C against G. And not just another step of our DNA ladder, right? And then we have our last nucleotide, and it's A. And we are going to put another T against it. So now we need to attach another rail. And I'm almost finished with this. So looks like a ladder, right? But DNA is not just a simple ladder. It is a twisted ladder. And I'm going to twist it. Look, this is what DNA looks like. And if you kind of look sideways, you might actually, see ladders this way much better. But this is what, a very simple representation of DNA looks like, right? Please let me know if you have any questions. Okay, so, and now we can move to our second activity, which is even more fun. And this activity is extraction of DNA, which means that we are going to take DNA out of strawberries. And I already have some strawberries right here. And these are the other reagents that we need. Just some dish soap. We also need some salt. Just the table salt would be good. We would need some water, right? And I already showed you strawberries, but it's good to take another look at them. And they need to be in a sandwich bag. And you'll see why just in a moment. And we also need a cup. And I'm using a cheesecloth, just, like a square piece, like this will work. And there is also some alcohol, right? And it's important that it's frozen, so I kept it in freezer overnight. Okay. So we are going to start by making our extraction mixture. And for that, we need to use water. And we are going to add a teaspoon of salt. And let's mix it up. And we also need to add some detergent, which is our dish soap. Tablespoon should be good. So this is our extraction mix. And now we are going to prepare our strawberries. So I've got like 3 strawberries, and I'm going to mash them up because we want, all the cells to be exposed. And while I'm doing this, just like that, right? I'm going to tell you a little bit about why we are using strawberries. Strawberries are delicious, and pretty much like every living organism, they have DNA. But a special thing about the strawberries that are sold in supermarkets is that they've been specifically selected for their high DNA content. So normally cells only have one copy of their genome, and those strawberries that we usually buy in the market, they have like 8 copies of what the wild strawberries would have. This is probably makes them so big and flavorful, but it also makes them really great for purifying DNA, because just think about it, you can purify 8 times more DNA than you would purify from some other fruit, which is really great. And remember, we really need to make sure that it's kind of mashed really well. It means that we will be able to purify more DNA, and that our procedure will be more efficient. So the way it's done is that we have pretty much like roughly the equal volume of strawberries and the extraction mix, which we just made, right? So I think, yeah, I've done a pretty good job at mashing strawberries. So now let's do some extraction. I'm opening the Ziploc bag and I'm going to pour the extraction mix in there. So we have plenty of strawberry cells floating around. And because we have some detergent in our extraction mix, the dish soap, so the cells will be easily destroyed. The membranes, they can stand detergent around, so which means that the DNA will be released from the cells, right? So yeah, this is the extraction process, right? We are helping cells to release the DNA. But we still have lots of, broken cells in here. It still looks pretty mushy, and we want to get kind of a clean and nice, clear liquid. So we are going to filter this mix, right? And I'm going to use a cup and funnel, and I'm also using the cheesecloth, which will help me, get rid of bigger chunks of strawberry. So I'm putting the cheesecloth right in the funnel and in the cup, and now I'm pouring my mix in. Right, you can see that there is a flow of liquid, and just to speed it up a little bit, I'm going to, kind of pick up the cheesecloth. So what we get inside are just, bigger chunks of strawberries. And what is going through the filter, through the cheesecloth, is some liquid which contains released DNA. Yes, I'm going to squeeze it a little bit more, just, to make sure that we get as much DNA as we can. All right. So, I'm just going to show you the cup. It has some, it is labeled, and you can see that we've got around 100 mils of this mixture, right, of this extracted DNA. But it's still like kind of, floating in this solution surrounded by salt, water, and detergent. And we need to do something to be able to see it, right? And what we are going to do, we are going to use some alcohol at this stage, ice cold alcohol, which was stored in a freezer overnight. And I'm just going to add pretty much an equal volume of it to this mixture. Okay. So you can kind of see that the mixture pretty soon it starts kind of separating itself, and you get some alcohol sitting on top, and you get our extraction mixture at the bottom. You can probably see it looks kind of whitish because there are lots of air bubble. We've been mixing, the room temperature solution with ice cold alcohol, which is, not surprising. But like if you look at the top of it, or if you like even look to the side, you can see formation of something really interesting. And this is our DNA, which, I'm trying to fish it out now, right? Which looks approximately like this. Some people say that it looks a bit like a snort, but yeah, you can say that, I guess. But the important part of this experiment is that this DNA was precipitated with alcohol. So what happened is that all the separate molecules of DNA, they kind of, got attached to one another, and we've got pretty much all the DNA, instead of floating, it kind of became solid. And this is why it's called precipitation. Okay? So once again, please let me know if you have any questions. And yes, this is just another explanation of what was happening when we had when we've been using cheesecloth for filtering. Right, so the bigger chunks of cells, they would stay inside of the filter, and DNA and smaller molecules would get through. Yep. So we had soap, salt, and alcohol. This is what we used for this experiment. And I kind of mentioned already about what's happening, but let's go over it again. So when we were using soap, we were breaking cell membranes. So we were destroying the cell so the DNA can be released. And then we were using salt, just table salt. And salt is important so that it kind of keeps DNA not in a separate chromosomes, not in a separate molecules, but it kind of helps it stick together so that it's easier to purify it later. All right, so now we've got some question time, and please remember, if you have any questions, put them in the chat and I will answer them right away. But right now, we'll try to kind of think over what we've seen. And I'm gonna ask you a question, think about it, and then we'll discuss the answer, right? So, what does DNA look like based on what you've seen today, right? Let's think about it. So, here is the answer that I've got. So, under the microscope, DNA looks like a twisted ladder, just like this one. This is what DNA molecule looks like. But when we purify it from, the actual fruit or, other living organism, it looks like a snort. And, this snort, it contains lots and lots of those DNA molecules. Well, that's a bit of a trick question. Did you eat DNA for breakfast, lunch, or dinner? Okay. So, most likely you did have some DNA, right? Because most of the food is made from the living organisms, and quite likely it has some DNA in it, just like, the strawberries that we used for DNA extraction. So, yes, DNA is a fairly healthy and quite common part of our diet. So the question about the extraction of DNA from strawberries. So why did we mash the strawberries? Why did we mash the fruit? Okay. So when I was using like Ziploc bag to mash the strawberries, I was trying to break the fruit into smaller pieces so that more cells are exposed to the detergent, and at the end more DNA is released. Another way of thinking about it is, to think about more surface area, right? When we only have like strawberries, we only just unmashed strawberries. We only have that much of the surface, but once we mash it, every little piece can be exposed to the detergent, which makes this procedure much more efficient. Okay. And next question is, what did the soap do? So the answer is soap or detergent. It broke down cells, their membranes, and as a result, DNA was released in the solution, and we could purify it later, which was pretty cool, I think. What did the salt do when we were extracting DNA from strawberries? Why is salt important? Okay, the answer is that salt helped DNA stay together throughout the procedure. So instead of, individual chromosomes or individual DNA molecules just floating around, they've already been kind of herded into small groups. And when it came to precipitation, it was, much easier to get them all together, right? Why did we use coffee filter pantyhose? In our case, we used cheesecloth, right? So think about why we used cheesecloth for DNA extraction. Okay. So the answer is our piece of cheesecloth, it served as a filter. It has a lot of small holes that only allow small molecules like DNA or water to go through, and the larger chunks of strawberries stayed inside. Next question is, why did we use alcohol? Okay, so the answer is that alcohol separated DNA from other filtered cell components, and this process is called precipitation. Basically, it means that DNA from the liquid form, it became solid, which means that it formed precipitate, right? But there was another question, why did the alcohol need to be ice cold for this experiment? And to be honest, I don't know the exact answer to that. Uh, I never tried it with room temperature alcohol, but I am assuming it makes precipitation works better. Okay, and this is it. We still got some time for questions or comments. And thank you so much for having us in your classroom.
That was amazing, Ghana. Thank you so much for presenting that. That was so much fun to watch, and I'm definitely craving some sweets after that first experiment for that first diagram. I'd also like to thank everybody else who is viewing for coming. We look forward to having you at future workshops. If you have any questions, the floor is open. And we'll be here for another couple of minutes. For any future workshops, you can check them out on our Eventbrite if you haven't already. We have experiments and workshops going on all week. And thank you again, Ghana. This was so much fun to watch.
Yeah, it was so much fun to do. Thank you for inviting me.
I think it looks like we don't have any questions. We have some thank yous. So again, once again, thank you so much, Ghana. We'll close off the broadcast, and this live stream will be live on our YouTube channel. It'll be posted after this ends. So if you want to share it with your friends and try the experiments at home, please feel free and send us a picture of what you make. Thank you again, and we'll see you at the next one.
Okay.